Everyone, my name is Humza Hemani. I'm a second-year PhD student at Washington University in St. Louis in the Muller Lab. I'm excited to present my talk today on studying the epigenetic regulation of the epithelial to mesenchymal transition in breast cancer. Trying to see if I can get a pointer here. Okay, I think I might have. Okay, all right, thanks. So we were interested in studying we wanted to study the interaction between signaling proteins, DNA, and other biomolecules after treatment of breast cancer cells with growth ligands and cytokines. Specifically, we wanted to study the onset of protein histone signatures and the resulting activation pathways leading to cellular phenotypes in response to these growth ligands. These ligands are a crucial part of how the tumor microenvironment modulates breast cancer progression, but the specific interactions and pathways involved are not fully understood. Why is it not going forward? To study this, the LINX Consortium project was created where MCF10A cells, a model for epithelial breast tissue, were treated with various growth ligands and cytokines and assayed for several different modalities. In this talk, I'll be focusing on the reverse phase protein array data to measure protein expression and the global chromatin profile data, which is a mass spec method to study histone marks or histone modifications. Again, the goal is to get an in-depth characterization of how epithelial breast cells respond to growth-promoting ligands, and therefore how the tumor microenvironment modulates these cells. To do this, we needed a mathematical model that allows us to model these different modalities of data together so that we can extract the onset of regulation signatures and the pathways involved using a systems approach. And so we created HOCMO, the higher order correlation model. This is a highly interpretable and self-supervised learning method to study complex inter and intra-pathway cellular signaling networks. To summarize this method, the reverse phase protein array data is, which we use to measure protein expression, is modeled as a tensor, as is the global chromatin profile data, which we use for histone marks. These are represented, these are their own tensor each, which we then decompose to get latent factors. These latent factors represent higher order signaling between signaling pathways. While our project used 2 data modalities, this method can be extended freely to 3 or more modalities. So the slide advances separately on this screen and that screen, apparently. If we take a look at the latent factors we extracted after applying HOCMO on our data set, we can see the strength of the regulation of protein expression and histone marks by each ligand at each time point. We can see the onset of regulation of these proteins and histone marks occurring at the 24-hour time point and going on to the 48-hour time point. This then gives us information on the timescale at which ligands present in the tumor microenvironment modulate breast cancer cells. If we look at the histone mark data, we see that the most significant regulation occurs with interferon gamma at the 24-hour time point, suggesting the potential onset of breast cancer initiation. HOCMO can also provide much more detailed and zoomed-in information on the pathways being regulated by these ligands. For example, HOCMO predicted the correlation between cytokine interferon gamma and the histone modifications H3K4 acetyl, H3K4 monomethyl, and H3K4 trimethyl. While H3K4 monomethyl and trimethyl are well studied in the breast cancer context and are associated with active enhancers and promoters, much less is known about H3K4 acetyl in the breast cancer context. HOCMO was able to identify the importance of this relatively unknown mark in breast cancer. Furthermore, if we look at the RNA sequencing data, we also see the downregulation of CDH1 upon treatment with interferon gamma. The specific change, the downregulation of CDH1, is a hallmark of a transition known as the epithelial to mesenchymal transition. And so the previous speaker actually mentioned this transition. It's key in breast cancer. Breast cancer cells must undergo this transition before metastasis can happen, and during it, they lose cell-to-cell junctions and acquire motility. CDH1 is a key protein in cell-to-cell adhesion, and so the loss of CDH1 is a key part of this transition. If we look at the RNA sequencing data and the cyclic immunofluorescence data, we see that alongside this loss of CDH1 expression, we have gains in STAT3 expression, as well as gain in ZEB1, which again was mentioned in the previous talk. suggesting that these proteins are involved in a regulatory network with each other. If we look at the CDH1 promoter, we can see that in addition to this increase in RNA expression of CDH1 here, it's shown in the top two tracks, or sorry, loss of RNA expression of CDH1, we can also see loss of accessibility of the CDH1 promoter, which we assayed using a TAC-seq. This then allowed us to create our hypothesis. We saw the increase of STAT3 expression in the RNA-seq data, and we hypothesized that STAT3 is binding to the ZEB1 promoter, which then goes and binds to the CDH1 promoter along with a histone deacetylase. We then hypothesized that this leads to the loss of the H3K4 acetyl mark, closing up of the chromatin, and therefore loss of CDH1 expression. To test this hypothesis, we use the cut-and-run assay. Cut-and-run is a relatively new assay which allows us to measure transcription factor binding and histone modifications, similar to ChIP-seq. Compared to ChIP-seq, we can use fewer cells to get higher signal-to-noise ratios. Looking at this data, we can see a clear binding of ZEB1 to the CDH1 promoter. If we look at the H3K4 acetyl mark data, however, the story is a bit more complicated. Again, so we see this loss of CDH1 expression and the loss of this accessibility of the CDH1 promoter, as I had mentioned. Upstream of the promoter, we see downregulation of the CDH3K4 acetyl mark, whereas we see increased enrichment of this mark at the promoter and shortly after it. Taking these data together, then, we see clear binding of ZEB1 at the CDH1 promoter and modulation of the H3K4 acetyl mark. The exact role of this modulation of the mark and how it interplays with the closing up of the chromatin, which we were able to see in the TAC-seq data, and how that then affects CDH1 expression remains to be studied. This study ties in with broader work done on histone modifications in breast cancer. This paper, for example, shows that the H3K4 acetyl mark domain is seen at the promoter regions of a list of key genes involved in breast cancer, including in different breast cancer subtypes. We also see different transcription factor binding occurring at introns in the CDH1 gene during the mesenchymal epithelial transition, which is a related transition also involved in breast cancer. To conclude then, Hawkemo was able to extract the onset of the regulation of protein processes and histone modifications at the 24-hour time point, and was also able to show the onset of specifically the epithelial to mesenchymal transition in response to interferon gamma, which we were able to validate using several assays. We identified additional histone modification signatures that suggest interplay with pathways related to the mesenchymal to epithelial transition. Next steps in this project include studying the heterogeneity of the breast cancer cell response to interferon gamma using single-cell multi-omic methods. I'd like to acknowledge the help from my fellow members in the Mulla lab, as well as our collaborators in the Roshman lab, GTAC, and the Heiser lab. Hawkemo is available now as a PIP package. Thank you.
Thank you very much. Do we have a question for our speaker? Yeah, with just one question. This is a very interesting data set, also especially with the association that you find specific markers. Are you planning to use some modeling data to see how the behavior of your genes work together?
Yeah, so this project does naturally tie in with these sorts of modeling projects. I think at the moment we're interested in going into the single cell level of studying this, but again, the modeling would be a very interesting sort of approach to take as well, of course.